Well, that's the toughest part of our job, and it's a multifaceted, multi-layer type of dissection, and it starts out with, this year we had almost 200 letters of intent, and out of those letters of intent, they get reviewed by the Scientific Medical and Advisory Board, and from that we get scores and comments, and out of 200 or so of these letters of intent, we will invite around 50 to submit what we call a full proposal. And that goes from like a one-page research summary in the letters of intent to 5 or more pages plus additional information that a review committee will then evaluate. So those get copied and go to a review committee consisting of 12 scientists. And it is our responsibility, Carmen and I, to match the expertise of those review committee members to that grant or that proposal being that's been submitted.
The trick is making sure you understand what the scientific content is and what the intent of the grant is, and match the expertise of the various members of your panel to what the primary aims and experimental objectives are of the grant. And it's nice having a mix of like basic researchers and clinicians in the room, as well as perhaps assign grants to a combination of basic and clinical, so that you understand all the aspects of it. With these grants in particular, we try to be sensitive to the significance and impact. In other words, we sometimes see some grants from a scientific point of view are very interesting, basic research, etc., but they're really not hitting the mission as we understand it. And so having the clinical side represented as well as the basic science is very important. It's really just knowing the panel members, knowing what's in the grant, and doing the best job you can to match the two.
And that's kind of part of our job, is to build up a committee that matches the best science relative to what we believe will be the proposals that year. And doing research for the goal of getting a cure is a step-by-step process. It's building blocks upon blocks upon blocks. And what we may be funding at this level is one of the very early blocks of, a brick block in a building, but that building in the end will be the cure. And it takes all those individual blocks to get to the top. And you can't do it, it's a kind of a linear process. You can't understand what's going to be at the cure until you understand some of the very basic processes. And that takes stepwise, slow, stepwise progression. We're making some very good progression, but still, we still got more blocks to put into that building.
Dr. Bell?
I tend to draw an analogy between, say, mitochondrial disease and cancer. Mitochondrial disease is not one disease, so there's not going to be one cure. Cancer is not one disease, it's a class of diseases. There's no one cure for cancer. There are treatments, however, and in the case of cancer, years of research, going back actually, more than 100 years of research, have led to in some cases, very effective treatments for certain types of cancers. And I think what we could hope for is that as we understand mitochondria's role in the cell and in the physiology of us better and better, we'll have a better shot at getting cures for the different types of diseases. But the funny thing is,
or targeted therapy.
Yeah, targeted therapy. Yeah, cure being different types of cures. Even something that is something that's a treatment that leads to better quality of life. But it's funny. I've been in mitochondrial research since my graduate days 40 years ago, and the complexity of the interaction of mitochondria in the cell has really only been started to be understood within the last, I'd say, 10 to 15 years. Yeah. For a long time, the mitochondria were thought of as autonomous parts that were sort of separate from the rest of the cell, and we really know that's not true now. And so that's what's leading.
We're dealing with new science, basically?
We're dealing with a better and newer appreciation of what mitochondria can and are doing. And like what Carmen was saying, for many years, mitochondria were one of the very early enzymology pathways that were studied, with the breakdown of sugars by Krebs and way back in the 30s and 20s. And then the genomic era started happening, genes sequencing and cloning and stuff. And the mitochondria were kind of pushed aside, saying, well, that's not exciting because we already know what it does. But that's Then came in '88 the first identification of a point mutation in mitochondrial disease, quickly followed by deletions. And now we know that hundreds of point mutations and deletions in the mitochondrial DNA-caused disease. The genomic era has supplied us with nuclear mutations, and that's as many as 1,000 gene targets.
So we're learning that mitochondrial mutations, deletions, abnormalities play a much larger role in disease than we once thought.
Definitely, definitely. I mean, we see it in Parkinson's, Alzheimer's, these deletions. Autism is one of the new ones on the block, but certainly not new by study because it's been described even as much as 10 years ago. Your money is the most precious money I can think of. NIH dollars and government-funded dollars comes from everywhere, every taxpayer. So it goes to thousands of different sources, but they're collected from taxes. This money from bake sales, from fund runs, is the most precious money we'll ever see. And so it makes our job harder. We just don't give it out freely. And so we want to find the best place to place your money, the donations, because it's a very precious source. It's an analogy of saying, if you want money from the UMDF, you have to go up to that girl or boy running that lemonade stand and say, can I have all your profits?
Interesting, interesting.
Yeah, and I would just add that the one thing you know with the UMDF process and the panel, the review that we go through, it's always being handled by people who are experts in mitochondria, be it basic research or in true translation in the clinical side. In the case of NIH and larger organizations, these grants can be reviewed at any one of dozens of different study sections, and oftentimes they don't get the attention they deserve. What this foundation does with its special funds, albeit it's not a lot of funds, it's significant. It is significant. You can help to seed that pilot data, those early research projects, those new investigators with new ideas that might get overlooked at NIH and really trigger something that could turn out to be important. And I think we've had several examples of that.
There's a lot of
important good things have come out of projects funded by the UMDF that probably would have been missed at NIH.
Well, it would be missed by NIH, and then having the seed money to get preliminary evidence so they can get an NIH grant. Now we've started labs that otherwise may have gotten shut down if it wasn't for UMDF money. And now they're up and running, they're independent, and they can get NIH funding, repeat or renewable funding, and carry on and offer an impact.